Okay, it's my pleasure to introduce the fourth speaker, who is actually the organizer of the symposium. So thank you for doing all that work organizing this. So the final speaker is Dr. Helena Gellersen. She is a research fellow at the German Center for Neurodegenerative Disease, or DZNE, and she's also at the University of Cambridge. So let's take it away. Let's give Helena a warm welcome.
Thank you so much for the kind intro. And yeah, again, thanks so much for the organizers for choosing our symposium. And I'm very excited to talk to you today about distinct contributions of dissociable brain networks and hippocampal subregions to memory fidelity. And I have no disclosures. Now, our normal experience in everyday life is characterized by a lot of feature overlap between different events. So for example, I hope a lot of us had hopefully had very nice birthday parties throughout their lives, but many of these will of course share features. So that means a lot of the neuronal populations coding for those two events would be highly overlapping. So this is, of course, confusing for the brain. So there must be some kind of system that manages to tease these apart, because otherwise we wouldn't know what are we exactly recalling from which event. And this term, or this process, is termed pattern separation, and it is crucial for high-fidelity memories whenever we have to discriminate similar events. And my colleagues have already done a beautiful work of introducing the entorhinal hippocampal circuit. But just as a very quick reminder, this is of course crucial for memory. And particular hippocampal regions are thought to be, in animal models and also some human work, shown to be involved in this pattern separation process, specifically via a circuit called the trisynaptic pathway, going from the entorhinal cortex to the dentate gyrus, which is characterized by very sparse neuronal coding patterns, which is thought to be essential in this pattern separation process, where you're decorrelating the neuronal activity patterns for two events. This is then passed along to the subregion CA3 and further to CA1 and then to the subiculum before producing a retrieval cue back to the cortex to reinstate that memory. So the dentate gyrus has often been called or has been proposed as a universal pattern separator. But this is actually really difficult to show properly in most human neuroimaging studies, because this region, oftentimes, these signals are overlapping with CA3 signals in 3 Tesla imaging. So we really do need ultra-high resolution imaging to tease these apart to determine, is it truly just the dentate gyrus, and is this region involved in a domain-agnostic manner? And as we've seen, the long axis of the hippocampus actually plays an important role in the heterogeneity of different signals. So these regions might not act exactly in the same manner. And again, we need high-resolution imaging to properly resolve this. And so far, we've heard a lot about the medial temporal lobe. But of course, there's a lot of other brain regions that are also engaged in memory. And specifically what we know, or what has been proposed, is that there are 2 networks in the cortex that are engaged in somewhat different processing. And this is the anterior temporal network for object information and the posterior medial network for spatial information. So a question is, how do these potentially contribute differently to information, to pattern separation for different types of information? And one of the reasons why this is important to determine is because these 2 networks are differentially impacted, for example, by aging and Alzheimer's disease. So by better understanding those, we might also get a better understanding into memory deficits in these conditions or in normal aging. And so my aims for this study were to provide a more fine-grained view of the contribution of different hippocampal subfields, the hippocampal long axis heterogeneity, and other medial temporal lobe subregions to memory fidelity, and pattern separation for different types of information, and to determine whether other regions besides the hippocampus exhibit pattern separation-like responses, and if so, whether this happens in a domain-selective or agnostic manner. And then finally, assess the behavioral relevance of activity of these various brain regions in explaining inter-individual variability in memory fidelity for different types of information. And a very well-validated paradigm that is oftentimes being used in non-invasive MRI studies to get at a pattern separation process are termed mnemonic discrimination tasks. So in these types of tasks, in the sequence, you always were presented with two novel items called targets, followed by either a similar law that is a new stimulus but shares a lot of common features, or the exact repetition. And we had an object task and a scene task version for this. And we scanned younger adults in a 7 Tesla MRI scanner, specifically focusing on the medial temporal lobe and parietal cortex at a resolution of 1 millimeter isotropic. And our regions of interest were manifold. So we segmented the amygdala, delineated anterior and posterior hippocampus at the disappearance of the uncus, so the hippocampal head and body. And then we manually delineated all of these beautiful hippocampal subregions. So a lot of work going into that luckily I didn't have to do myself. But here the crucial subregions that we're looking at are the subiculum, CA1, CA3, and the dentate gyrus. And then we used FreeSurfer to delineate the various regions of the posterior medial network and the anterior temporal network. And what we were here particularly interested in doing was to analyze all of this in native space to really keep as much of the inter-individual variability in the very complex anatomy of these regions. And just the first question was, can we actually recover these category-specific regions in these networks? And this was just a relative comparison of objects versus scenes. And you see this is very much overlapping with the canonical networks. So here, the posterior medial network very much strongly, relatively more strongly responds to scenes than objects, and the opposite is the case for anterolateral regions. And this also continues into the medial temporal lobe. So, for example, looking at the posterior medial entorhinal cortex and parahippocampal cortex more engaged during scenes, same with the subiculum, whereas BA35, so the perirhinal cortex and the amygdala, are relatively more engaged during the object task. So, of course, here we have the challenge that we cannot record from individual neurons. So what actually counts as evidence for pattern separation in an fMRI task? And what I did here was to characterize every region's activity profile across all of the different trial types. First, a region that supports memory should be more active during a first or novel presentation of a target than to its exact repeat. Importantly, a region that is involved in pattern separation should not reduce its activity strongly during a presentation of a correct law, because that brain region is capable of saying, no, this is actually a novel stimulus, even if there is feature overlap. And consequently, it should also be more active during a correct law than a repeat. And ideally, we would also want to see that this is being reflected in the behavioral outcomes, so that this brain area is more active during correct versus incorrect laws. What we can then further do is look at the degree of target-lure pattern similarity, or the similarity of different features that we find between these two stimuli. And what we would expect across similarity bins for a region that is not involved in pattern separation and more interested in complete novelty, this is the pattern that we would want to see. A very strict decline from a novel target presentation to even then a low similarity law, and then not that much further modulation as the feature overlap increases. But a region that is involved in pattern separation should maintain relatively high activity, even to laws that have a lot of feature overlap, because this region would treat all of these different laws as a new stimulus. And the way in which we can assess this is to characterize different lures into these different similarity bins. And we've done that by looking at an independent sample of over 1,500 adults throughout the lifespan to avoid circularity in our data, and then classified these lures into different similarity bins. And this is what I've then done. This is specifically for all of the MTL regions. And because our whole brain activity showed very clear hemispheric differences, I split this up by hemisphere and also into anterior and posterior subfields. And to guide you through this, so these are all of the regions as I just described. Down here are all of our pairwise comparisons of trial types. And as you can see here, most regions, as we would expect, show repetition suppression for an exact target repeat. Obviously, the MTL is strongly involved in memory, so that's exactly what we would expect from these regions. But very few regions actually show a pattern separation-like response. Specifically, the amygdala meets relatively many of these criteria for the object task. The anterolateral entorhinal cortex does so for both stimulus types. The posterior subiculum and posterior CA3 and dentate gyrus do so for the object, so really only the posterior aspects, and this was not as clear in the scene task, most likely because the pairwise comparison are FDR corrected, so this is a very, very stringent correction overall. And what is very striking here is that the anterior and posterior subfields have very different patterns of responses. So the anterior subfields, all of them treat a lure like a stimulus that has a lot of feature overlap and thus is not completely equivalent to a novel item, whereas all of the posterior regions have a greater tendency towards treating the lure almost like a novel item, so no significant difference to the presentation of a new target. If we further look at this with a curve fitting for these MTL responses, across, so what we're looking at here is a B-spline model with a knot at a low similarity would suggest this type of sensitivity to even low levels of feature overlap, as I've shown earlier. A linear model would be some kind of resistance to feature overlap, but in a gradual manner as feature overlap increases. And this is exactly what we see in the anterior subiculum for objects. So there's a very clear decline already, even at low levels of feature overlap, but a generally more resistance to feature overlap in scenes, which is consistent with the idea that among hippocampal subfields, the subiculum really is the one that is most domain sensitive or has the greatest domain preference towards scenes compared to other regions. And all other anterior hippocampal subfields, regardless of its CA3, CA1, or the anterior dentate gyrus, they all show this very clear preference towards complete novelty. So no direct evidence of pattern separation. But we actually have quite a different pattern for the posterior hippocampal subfields. Specifically, for CA1, CA3, and the dentate gyrus, we all have a linear pattern, so this suggests some modulation by and some degree of resistance to feature overlap. The posterior subiculum is a little bit more resistant specifically for the scenes, so suggesting that it treats a low similarity lure just as a novel stimulus. And interestingly, specifically the posterior left dentate gyrus is showing this exact pattern separation-like response that we would expect, but specifically for the objects only. And the anterolateral entorhinal cortex shows also a more gradual decline over similarity bins, and this is rather consistent for both of these types of stimuli. So this is really the striking response pattern, and this is all on the basis of Bayesian information criterion model selection, so the most stringent way of selecting the best fitting models. And we see a little bit of this evidence for the scene task in the posterior subiculum. But what about wider cortical regions outside of the MTL? Can we also see that? Indeed, for the angular gyrus, we actually don't see too much domain preference, even though this is what we have seen in the direct comparison of objects and scenes. And there is more of a pattern consistent with the idea of this region being more resistant to feature overlap. And the same is the case for the inferior temporal sulcus, but the other posterior parietal regions very clearly favor complete novelty. And this is reflected by their curve-fitting responses. Again, very clear novelty preference in all of these parietal regions, except for the precuneus, and the angular gyrus in the scene task. So we do see that potentially the right angular gyrus in particular is more modulated by these scene overlapping features and does somewhat potentially, even interestingly, boost its activity to the moderate lows. This might have something to do with memory difficulty trying to resolve this higher degree of interference, which then later on fails at the highest lower stage. And we have a little bit more of a gradual decline for angular gyrus in the left hemisphere, which is consistent with the idea of this hemispheric difference between spatial and object function. The final question was, is this behaviorally relevant and can directly explain inter-individual variability in memory performance? And indeed, this is specific to the correct low versus repeat fMRI contrast, that it is only the dentate gyrus that can do so for scenes. And the repetition suppression contrast for first versus repeats is a lot more widespread, but it is not as prescribed to specifically the AT or PM networks. And so here you see the scatter plots for that. And to determine to what extent these share variants or have unique variants that they contribute to inter-individual variability in memory performance, I used a stepwise regression to identify the best fitting model. And it is specifically the dentate gyrus and CA1 for the scene task that predict higher performance in the scene task for individuals who also show greater activity or greater contrast between these trial types. And for the object task, both PHC and the dentate gyrus show up. So it's only the DG that really is directly and in a relevant manner predictive of behavior across different domains. After FDR correction, this does not quite hold for regions outside of the MTL. So here we only have trend level associations in the angular gyrus specifically for the correct versus lower, and a bit more general in inferior temporal sulcus and angular gyrus for the first versus repeats. But once we control for FDR, for multiple comparisons with FDR, this is no longer as relevant. So what we can say is, despite category preferences that were relative when we're comparing the magnitude of activity to objects and scenes, many AT and PM network regions are involved in memory for both objects and scenes. There's a very clear distinction between the hippocampal head and posterior hippocampal body regions, with the anterior regions generally really preferring complete novelty, and the posterior regions being more resistant to feature interference. The anterolateral entorhinal cortex, amygdala, angular gyrus, and to an extent the inferior temporal sulcus all treat similar lows more like novel stimuli, but only when we're averaging across all similarity bins, so they do fail at the higher degree of feature interference. Only the posterior dentate gyrus activity fulfills all criteria for pattern separation in the object task, and dentate gyrus activity explains inter-individual variability in mnemonic discrimination in a domain-agnostic manner. So I'd like to thank the team at the DISA-NE and the funders, and we're now open for questions.